Okay, get the sheet with the essays on. We're gonna do the two big essays on the front. What I wrote on the whiteboard is not part of the essays. What I wrote on the whiteboard is so you can understand the essays. Everybody got it? What's also important to remember is that ganglion cells are basically just multipolar neurons. With several dendrites, well, in the foveus centralis is only one, but don't worry about it. It acts like a multipolar neuron. Several dendrites, cell body, one axon hillock, and that axon is actually cranial nerve 2, the optic nerve. Are we all clear on that? This is really important to have that down before we go over the essay. Now, let me just number these so these make sense. So what I have on the whiteboard is point 1, 2, 3, 4. Will that help? No?
Yeah.
Okay. First of all, let's explain the difference of the two forms of retinine that we covered there on Monday. So this is part of what we already covered. Remember when the retinine is in the dark, it is a slightly bent molecule. It's a form of fatty acid derived from vitamin A, and it's bent at the 11 carbon, cis 11 retinine. And when that's happened in the dark, it fully hugs the protein. In the case of rods, the protein is scotopsin. Okay, does that ring a bell from Monday?
Mm-hmm.
And when light hits it, the double bond changes to a single bond and it becomes transol retinine, straightening out and partially separating from scotopsin.
Mm-hmm.
Okay? So that's the first 4 bullets right there. And that starts a chain reaction. So let's talk about establishing the dark current. What you want to write there next to that, establishing the dark current, it is a current of sodium ion. Sodium ion is just doing a circle and a current. That's the dark current. Okay? All right. So we're done with the first 4 bullets, just establishing the two forms of retinine, where it comes from, what happens when light hits it. All right, so again, when there's no light hitting the rod, the chromophore is in the form of 11 cis retinine. It is fully associated onto all the relevant membrane proteins, which is scotopsin. When this happens, the levels of cyclic GMP, which is a signaling molecule inside the cell like cyclic AMP, very similar, because they're both purines. But the levels of cyclic GMP go up. This causes the sodium channels to stay open. So this is what is showing on this slide. There's an increase in cyclic GMP when the 11 cis retinine is hugging the scotopsin. This opens the sodium channel, and sodium just goes flying in. When this happens, it lifts up the membrane potential to 0. Okay? Now we've got to remember one, rods only do graded potentials only. It releases neurotransmitters based on the degree of depolarization. 0 is depolarized. Okay? So what does that mean? That means it is going to really release a ton of glutamate. The neurotransmitter glutamate is released in a constant flow. Because of point number 2. Glutamate has an inhibitory effect on the bipolar cells. Point number 3. This causes the bipolar cell to hyperpolarize, and it cannot release its excitatory neurotransmitters on the ganglion cells. Why? Because it's hyperpolarized. And point number 2 says it's going to release it only when it's depolarized. Is everybody following that logic? Do you see why you need these points to understand the essay?
Yes.
Well, if there is no excitatory neurotransmitters on the ganglion cell, it cannot reach threshold because the ganglion cell is a multipolar neuron. It's got to hit threshold here at the axon hillock. Well, it can't because it's not getting excitatory signals from the bipolar cell. That's why you got to know these cells in order. And that's why you don't see anything in the dark. If you remain in the dark a long time, the rod is going to maximize all the rhodopsin and put as many discs as it can in the cytoplasm. This is what we call dark adaptation. Monday Lab, does this ring a bell?
Yeah.
Wednesday Lab, you'll see that this afternoon. All right, any questions on establishing the dark current? So the dark current is this: because of the sodium-potassium pumps, the sodium is just going in and then out, and then in and then out. Do you see the circle of sodium? That's the dark current, and it gets to 0 millivolts. It depolarizes to 0. Okay, because it's supposed to be minus 70, right? Well, it's 70 millivolts higher than that. That is depolarized. Any questions? Think this through sequentially. Whenever I get lecture exam 2 essays, I can tell the students who have not gotten sequential thinking down yet. They're all over the place. They're mixing up which neurotransmitter comes from where. Glutamate comes from rods. Don't worry about the bipolar cell neurotransmitter because there are several. You don't have to name it. It depends where you are in the retina. It changes the neurotransmitter. Okay, now what happens when the light comes on? This is the bottom half of that sheet. This is from your textbook for the dark current. There's way more to this than that. If you want to look it up about calcium and everything else, go ahead. I'm telling you what you need to know for lecture exam 2. Okay? All right, what happens when the light comes on? When the light comes on, the 11-cis retinine is converted to transol retinine, partially separating from scotopsin. This is going to cause the cyclic GMP levels to drop here. That's going to cause the sodium channels to close. With sodium no longer diffusing in and the permanently open potassium channels, because all cells have that, causes a constant outflow of potassium out. The rod now hyperpolarizes. And because it hyperpolarizes, it no longer releases glutamate on the bipolar cell. If you do not have this inhibitory signal on the bipolar cell, it spontaneously depolarizes. And when the bipolar cell depolarizes, it now can release its excitatory neurotransmitters here, which allows the ganglion cell to reach threshold here. And you get an action potential on cranial nerve 2 all the way down the visual pathway I'm going to cover here in just a minute. And that's why you see when the lights come on. Did I cover everything on the second essay?
Yeah.
So you got to know that this coming out of the rod is glutamate. And that is inhibitory on the bipolar cell. This neurotransmitters, and there's several, the bipolar cell neurotransmitters is excitatory.
Okay.
Any questions? And basically what I drew there in color is what's on these 4 points. You see why you need the 4 points to understand the essay? Any questions? Just think it through sequentially. People do not make this class more difficult than it is.
The release of the neurotransmitter based on the degree of depolarization, does that have anything to do with the amount of light you're getting in or not? Like how much it unbends or bends?
No, well, again, dark adaptation is gonna make the rods more sensitive if you get more discs.
Okay,
So the amount of light is relative,
okay,
depending on how many discs the rod builds up.
okay.
Okay? Any questions? So this, again, what I just drew right here, this is inhibitory neurotransmitter glutamate. This is excitatory neurotransmitter on the ganglion cell. Everybody see that? Previous slide. This is inhibitory. If the bipolar cell hyperpolarizes, it can't release excitatory neurotransmitters here. Any questions? Okay. Then let's move on. Again, you don't have to worry about transducins or phosphodiesterase. Just keep it to this. This is complicated enough. Everybody got it?
Yes.
Again, this class could be a lot harder than the way I make it. All right, let's talk about cones now that we're done talking about rods. There are 3 different types of cones, red, green, and blue. Now they all 4 photoreceptors have the same retinine. The difference between the 4 photoreceptors, rods, and these 3 cones is the protein boulder. Scotopsin can absorb light from all the visual spectrum, but these proteins can only absorb certain wavelengths of light. This is why cones can give you information about color.
Oh, shit.
Okay? Erythrolate, which should be able to, like erythrocytes, red blood cells. Chlorolate, chloroplastic green. If your fingernails turn blue, you're cyanotic. Cyanolate. Can you guys remember? Find a way of remembering these colors. All right, now this is also the opposite of a very similar slide I covered on Monday on rods. This is just the converse. When we talked about rods, it said it had low threshold. It said it had high convergence, so it had low visual acuity. This is just the opposite paired content. What do you do with opposite paired content?
Learn one.
Just study one and know that the other is the opposite, so you don't mix them up. Cones, you need more light to see color. If you don't have enough light, everything's in grayscale. 2, now this is where I drew more dendrites here. If you have a bunch of rods all coalescing onto one ganglion cell, okay, everybody pay attention to this because I gotta get moving here. And I will leave you behind today if you're not engaged. If you have a bunch of rods all converging, and this is convergence, on one ganglion cell, if I have 2 points of light here and here, how many points of light does your brain see?
1.
1. That's low visual acuity. Everybody got it? Now with cones, you have less of this convergence. That's what it's saying there. Thus, 2 points of light are going to look like 2 points of light. High visual acuity. Everybody got it? In the fovea, you guys remember fovea centralis?
Yes.
In the retina?
Yes.
In the fovea centralis, there is 0 convergence, which means 1 to 1 to 1. In the fovea centralis, it's all cones, and it's 1 cone, 1 bipolar cell, 1 ganglion cell. 0 convergence. 1 to 1 to 1, which is why at the fovea centralis you have the highest visual acuity. 2 points of light can be really close together, your brain still sees 2 points of light. Way out in your peripheral vision, you can have 2 points of light way far away, and your brain is like, duh, I only see 1. Everybody got it?
Mm-hmm.
Okay. This is talking about the different wavelengths of light. When you talk about wavelengths of light, you're talking about the electromagnetic spectrum. And it goes all the way from gamma waves to radio waves. including microwaves and X-rays and infrared and ultraviolet. And our eyes see just a narrow slice of the spectrum. We are blind to everything else except for this narrow slice. And that narrow slice is 400 to 700 nanometers. Okay? White light is the whole spectrum from 400 to 700. White light is all the colors. This is upside down compared to this scale. You see that the small, short wavelengths are up on top, on this one they're on the bottom. But that's the color of the rainbow, is the visual spectrum spread out. Any questions? And so here's where the proteins, again, it's scotopsin for rods, erythrolate, chlorolate, cyanolate for cones. Everybody got it? That's the difference. They absorb different wavelengths of light. Scotopsin can go across the whole spectrum. But cyanolate absorbs longer wavelengths. Erythrolate, shorter wavelengths. And chlorolate absorbs right in the middle. And that is why we can see the whole rainbow. These are primary colors, blue, green, and red. And so they mix to form the rest of the rainbow. Your cones have to work together to perceive this rainbow by firing percentages in those areas of the retina. Since blue is on one side of the visual spectrum, you just got to excite blue cones and you're going to perceive blue. There are no yellow cones. Why can you see yellow? Because in that area where yellow is sitting in the retina, 83% of the red cones are firing and 83% of the green cones are firing. And when in that area of the retina, you get that percentage of red and green cones firing, your brain perceives it as yellow, even though you don't have yellow cones. Is that just wild?
Yeah.
Oh wait, I got more for you.
Okay.
Since green is in the middle, yes, green cones will definitely give your brain a perception of green, but if you notice, there are a ton of different kinds of green. And all of it has to do with altering these percentages. And that'll be the difference between forest green and olive green. Everybody got it? Any questions? If you get a chance, copy this website off and put it in your device, laptop or something. It actually is pretty cool how it shows how colors mix and how they are perceived in the brain. But here's something that I don't know if you knew. But color TVs were actually developed in the early 50s. By the end of the 50s, they had color TVs down. It's just they were so expensive, no house owned them. They were just really expensive. The cost of color TVs came down in the 60s, and that's where more houses got color TVs, was in the 60s. But it was around for 10 years before that. Everybody got it? Because of people actually paid attention to science back then. I saw this one meme that kind of cracked me up. You know, scientists in the 90s, you know, we just cloned a sheep. We've got a robot operating on Mars. Scientists today, for the last time, the world is round. That is actually very serious. Sad and true and tragic and funny. But back then, people actually paid attention to science, and because they paid attention to the physiology of cones, not only did the TV manufacturers, but those that made cameras to transmit color signals knew you only had to transmit 3 colors. And just alter the ratios like this on the bottom. So in an area that was yellow, 83% of the red dots would project, 83% of the green dots would project. Your brain would see it as yellow, even though there are no yellow dots. Everybody get it? Isn't science bitchin' when you actually pay attention to it? Yeah, the original color TVs was sending colored dots, and they were only blue, green, and red. And the reason why I made this slide is because NBC used to tout how they're broadcasting more programs in color with their peacock. And the peacock showed all the colors of the rainbow. Yet you're only projecting 3 colors.
Wait, what?
NBC was only broadcasting 3 colors,
Oh.
yet could show the peacock with all the colors of the rainbow by altering the ratios and percentages. Because they understood visual physiology of cones. Today we got HD TVs, they're transmitting all the colors. But back then, it was just 3. Everybody got it?
Mm-hmm.
Was that interesting?
Mm-hmm.
All right, let's now do this visual pathway. I just got it started here. Go ahead and feel this out. I just have the basic sequence here on the whiteboard, but there are other cells in the retina. Amacrine cells and horizontal cells. You only have to know about horizontal cells. I'm not going to ask you any questions on amacrine cells. Okay, so photoreceptors go to bipolar neurons. Bipolar neurons are going to go to ganglion cells, but before I do, that's why I'm talking about horizontal cells. Horizontal cells do this thing called lateral inhibition. Lateral inhibition is taking out signals that are weaker so that only the strong signal goes through. This is a form of refinement that neurologists recognize as a CNS process. Only the central nervous system does lateral inhibition. What is in the CNS?
Your brain.
Brain and spinal cord. Does your retina look like brain and spinal cord? And this is why neurologists consider the retina to be part of the brain. Because it does lateral inhibition with horizontal cells, it must be CNS. Everybody got it?
Well, do you think it's part of it?
What's that?
Do you think it's part of it?
Hell yes. But I went to school a long time to learn the complex realities of the body. So this doesn't bother me.
Is it just because the cranial nerve is connected to the brain? Is that why it's
No,
doing it? Or?
it's because this refinement is not something that happens in the peripheral nervous system. So, okay, if it's just brain and spinal cord, retina's gotta be part of the brain. Any questions? All right, this lateral inhibition is really, and this is what I'm gonna already mentioned already, but I'm gonna bring it home again and again and again. This lateral inhibition is really crucial outside the fovea centralis. You need this refinement when you have convergence. If it's 1 to 1 to 1, you don't need lateral inhibition. That's why there are no horizontal cells in the fovea centralis. outside the fovea centralis only. To give you an idea, at the periphery, you can have over 100 photoreceptors, there'll be all rods out there, going to one ganglion cell. You can have points of light in over 100 spots, and your brain will only see one point of light. And that's why out here in the peripheral vision, you can't hardly see any detail. Does this make sense? You gotta get it right centered where it is 1 to 1 to 1. Then you have visual acuity. But if you have that kind of visual acuity, you don't need horizontal cells with lateral inhibition. All right, let's continue on down to the ganglion cell. The ganglion cell is a multipolar neuron. Now, when we talk about receptive fields, and I would write this next to this top box, this is outside the fovea centralis. You don't really have receptive fields in the fovea centralis because it's 1 to 1 to 1. But where you have convergence, you're going to have these things called receptive fields. Where you are outside the fovea centralis, you have two types of ganglion cells, on center and off center. They are completely paired and opposite. So what are you going to do?
Learn one.
Just learn one and know the name of the other, and know that it's opposite. So I'll do on center. On center, and again, you have to have convergence. So you have to have several photoreceptors coming into one ganglion cell. You have to have more light at the center, and that's going to cause depolarization. Well, depolarization of the bipolar cell. Don't worry about the photoreceptor. And when you get more excitatory neurotransmitters, when the bipolar cell depolarizes, this is going to reach threshold. You're going to get an increase in action potential firing rate. So for an on-center ganglion cell where you have convergence, if light hits the center, you're going to get an increase in action potential firing rate. For those ganglion cells, if light hits the periphery of the receptive field, it's going to hyperpolarize. You're not going to get action potentials, or very few. The off-center is the opposite. Just learn one, then learn the name of the other. Okay? This form is also synaptic integration, which is also a CNS process, synaptic integration. So, again, if you are an on-center ganglion cell, so this is several photoreceptors in this circle. Everybody look at this, and you should write this down. Several photoreceptors in the circle converging to one ganglion cell. That does not happen in the fovea centralis, which is why on the last slide I had you write outside the fovea centralis, where it's 1 to 1 to 1. This is not 1 to 1 to 1. So if you have light hitting the center, more action potentials. Everybody got it? Light hitting the periphery, it's going to hyperpolarize. You're not going to have action potentials, or very few. Off center is the opposite. Do you see how there's convergence here? You have to have convergence to have these types of ganglion cells outside the fovea centralis. This is just the same thing. Different pictures of the same thing. You can find this on the internet, it shows you the same thing. Okay? Again, the axon of the ganglion cell is the optic nerve. That's why they all have to gather at the optic disc to exit the eyeball as the optic nerve. Again, just learn one, the other's the opposite. Any questions? Okay. This is also now outside the fovea centralis, but this is about cones. So write down next to this slide. This is outside the fovea centralis, but this is only about cones converging. And by the way, if there's convergence onto a ganglion cell, they are all rods. If it's outside the fovea centralis and you have convergence of cones to a ganglion cell, they're all cones. You never have a ganglion cell getting both cones and rods. Everybody got it? Everybody got it? Okay, the convergent patterns for cones onto one ganglion cell. If they're both excitatory, we call that the single convergent pattern because they are both excitatory. This is going to give your brain information about color brightness. How bright is that red? If one is inhibitory and one is excitatory, it's called an opponent convergent pattern. And this gives your brain information about the specific color. What color of the rainbow is it? That's opponent. How bright is that color? That's single. All right. Again, all the axons and ganglion cells gather at the optic disc, exits the eyeball and the optic nerve. Then it's going to go, and it's the same axon, it's going to go optic chiasm, optic tract. And it's the optic tract that enters the brain. What you need to know is that the optic nerve has all the signals from a single eyeball, but the optic tract has signals from both eyeballs because some of them cross at the optic chiasm. Not all, but some. So on either side of the brain, you're getting information from both eyeballs, and it's going to split once it enters the brain. So let's just call this the brain line here. And about 1/4 of it is going to go to the superior colliculi. It's much more complicated than that. But this box here is about 3/4 of it is going to go to the lateral geniculate body of the thalamus. All senses synapse in the thalamus. For vision, this is where it synapses. Lateral geniculate body. Since you have one on the left and one on the right, and each is getting information from both eyes, this is where depth perception is processed. Okay? By the way, close one eye. Look straight ahead. Everything is on the same depth. The only way your brain can get information of what's front and what's back is you have to move your head. And when you move your head slightly, things in front move more, things in back move less. Now your brain can figure out what's front and what's back. If your head is not moving, and you only have one eye open, you cannot tell what's front or what's back. Your brain is generating illusion of what's front and what's back, but it is entirely an illusion. If you have both eyes open, you can tell without moving your head what's front or back. That's the depth perception. Okay, so the short arrow is 3/4. The long arrow is about 1/4, going to superior colliculi for visual reflexes. From this, you get this huge spray to the back of the brain called optic radiations. And it's going to head to the occipital lobe. primary visual area of the occipital lobe. That's the visual pathway. This is not or, this is optic radiations. Okay, I'm not gonna write it out. You got it, words out in the slides. Okay, any questions? Okay, vision is a completely complex illusion. Here's where the depth perception gets figured out, when your lateral pediculate bodies gets information from both eyes on either side. This is a little bit of the complexity. When you get to the occipital lobe, that's where this is, occipital lobe. There are different types of neurons found in the occipital lobe. You have simple, complex, and hypercomplex. Because your occipital lobe is only receiving action potentials. Some are about the edges of things. Some is about angles and rounded corners of things. Some is about movement and rotations of things. And you have to have different neurons that can generate that illusion from action potentials. Because your brain sees nothing. Everybody got it? Right. And then the visual association area is where you interpret what you see. And for you guys, this is where the information goes from your occipital visual cortex to the limbic system. It's got to process through the visual association first. You've got to make sense out of what you see, or read, or draw, or outline. If you're looking on the whiteboard, you got to make sense of it. Once it makes sense, now you can send it into your limbic system and remember it. It's very difficult to get it into your limbic system if it has not made sense in the pink box. Everybody look at me. The pink box is where active learning is going to make sense of the content. Like this. Oh, going connecting the dots from the rod photochemistry to the visual pathway. Can you see it on the whiteboard?
Mm-hmm.
Well, that's just this box, if you can see it on the whiteboard. If you have not engaged it actively, it doesn't make any sense to you. And then nothing goes into the limbic system. Good luck. Make it make sense. Now this is the way my brain makes sense of things. You gotta find the way that your brain works, because it may not be the same as mine. All right, any questions on
My brain's good.
vision? We actually only have one more lecture after this, and lecture exam 2 material is done. Are you already seeing how this depolarization, excitatory, hyperpolarization, inhibitory is in every lecture? Mm-hmm. Remember that one slide I said, you better have this down, and this has got to be as natural as breathing. Because it is in every lecture of lecture exam 2. And in fact, it's going to be here on when I talk about the ear. We are going to concentrate mostly on the inner ear. Remember, you have external ear, middle ear, and inner ear. Do you remember that from anatomy?
Mm-hmm.
Remember that the ear is loaded with threes.
Yeah.
There are threes everywhere.
Yeah.
Like external ear, middle ear, and inner ear. 3. Then let's go into the inner ear. There's 3. Cochlea, vestibule, semicircular canal. There's 3. How many semicircular canals do you have on each side? You got 3. How many chambers do you have in the cochlea? You got 3. You guys see how there are 3s all along the in the ear?
Yeah.
Okay.
I wasn't a fan of the ear.
External ear, middle ear, and inner ear, we're gonna concentrate mostly on the inner ear. When we get to hearing, I will talk about the middle ear just a little bit. We're gonna be mostly in the inner ear. And there's the three sections. Cochlea, vestibule, semicircular canals. Remember that the entire inner ear is in a bony shell, technically part of the temporal bone, called the osseous labyrinth. Paralleling on the inside is a membranous sac. This is what I drew on the whiteboard Wednesday lab. You'll see it this afternoon. There are fluids in the inner ear. Between these two labyrinths is perilymph. Chemically, it's very similar to cerebrospinal fluid, by the way. But inside the membranous labyrinth is endolymph. And it is a freak. Why? Because it's an extracellular fluid, but its contents is very similar to intracellular fluid. One of the few ones that's like that. It's freaky. All right. Now we got to get into the physiology. Go ahead and fill in the blanks here. Okay. All three areas of the inner ear have hair cells. Okay, they aren't hairs, they're cilia. There's two different types of cilia. There's one long one called the kinocilium. And several smaller ones called stereocilia. We're going to look at that in just a second. The cochlea doesn't have a kinocilium, it only has stereocilia only. All three cilia, for all three regions, are embedded in jelly. In a semicircular canal, it's going to embed in the cupula of the crista ampularis. And the vestibule. The jelly is the slopy membrane called the otolithic membrane. And in the cochlea, it's a jelly-like patio roof. Looks like a patio roof. And it's jelly-like, and that's the tectorial membrane. That's the jelly that the cilia are stuck in. And then the location is right here. Actually, I should look at this over here. So all three regions of the inner ear have some similarities. They have hair cells with cilia embedded in jelly. You just gotta know where they're located. Okay, so this is basically where they're located. The physiology is that if you bend the cilia, it's gonna convert it into an action potential firing rate. And maybe you should write that up here near the top where it says fire action potentials when bent. It's going to fire anyway. You're going to alter the action potential firing rate. So here are the two different types of cilia. The one long one with the club end on the ends, that's the kinocilium. The stereocilia are the shorter ones. They look like hairs. That's why they call these hair cells. Again, stereocilia is in all three regions, but there are no kinocilium in the cochlea, only stereocilia. So you got that one long one with the club ends, kinocilium, and then you have the stereocilia next to it. Look at this. Gelatinous layer. All three regions, the cilia is stuck in jelly. Are we clear?
Mm-hmm.
All right. Here's some more of this. Depolarization, hyperpolarization. Now we're just talking about, in fact, you should write this down here underneath this left column. Write action, potential, tone. When the cilia are straight up, you still have action potentials, but it's firing at a resting action potential tone or frequency. It is still firing at a resting action potential frequency or tone. And you can see it right here. Boom, boom, boom. Each one of these spikes is an action potential. Boom, 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 boom. Resting, tone, still, firing, regular rate. Boom, 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 boom. Everybody see that? Mm-hmm. Now what happens when you bend it? If you bend the shorter stereocilia toward the big club, or bend the big club away from the stereocilia, all meaning this direction, it depolarizes and you alter the action potential firing rate. Boom, 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 because it's depolarized. Bend it the opposite way. Bend the stereocilia away from the big club, or bend the big club toward the stereocilia, and you hyperpolarize the hair cell, and you alter the resting tone to boom, 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 boom. Everybody understand what's going on in this picture? Mm-hmm. You see how depolarizing excitatory and hyperpolarizing inhibitory is still all the way to the end of this lecture exam 2 material. Mm-hmm. And these cells can fire action potentials really fast. Again, baseline, depolarized, hyperpolarized. You alter the tone. This is paired opposite content, isn't it?
Yeah.
What are you gonna do?
Learn the one.
Just learn one, know that the other's the opposite. Okay? All right, let's now go to the vestibule first. This is referred to as static equilibrium. These terms are oversimplified misnomers. Don't worry about it. The reason why we call this static equilibrium is because you can alter the action potential firing rate by just altering your body position. That requires gravity, by the way. No gravity, body position don't matter. You should write that down next to this. Body positions require gravity. The utricle detects horizontal accelerations and decelerations, kind of like stepping on the gas or stepping on the brakes in your car. Horizontal, on a horizontal road. Saccule is going to detect vertical accelerations, like when you're riding in an elevator. And as I wrote there in blue, for the vestibule, the jelly is the otolith membrane, or otolithic membrane. Otoliths, or statoconia, those are synonyms, are rocks of calcium carbonate, and it gives the membrane some mass for inertia. And again, all you have to do is tilt your head, and you can make this otolithic membrane just sloop one way or another way. Everybody got it? But it requires gravity. You don't have gravity, this slooping doesn't happen. Okay? And it's located in the maculae. It's one for the utricle, one for the saccule. And this is talking about the otoliths or statoconias. It's going to increase the mass, increasing the inertia. This is what gravity is grabbing onto. Because remember, gravity is mass dependent. Like when we were doing filtration, you guys remember that in one of the first labs?
Mm-hmm.
Well, here's the weird thing. Look at this picture. All you gotta do is tilt your head down, and you can get the otolithic membrane to sloop forward. Everybody see that? Does everybody see that? It's bending the cilia when it sloops forward. This same slooping happens when you press on your brake. Just goes forward. And that is the physiology of Star Tours. Motion sickness, now there is all different degrees of motion sickness. Motion sickness can happen just from too much sensory input for some people. For most people, it is when the sensory input into your brain from the 8th cranial nerve from the inner ear does not match with the visual signals coming in from cranial nerve 2. When the two are not matching, your brain can't handle it, mismatch. Tries to make sense of it. Oftentimes people will get motion sickness. And it is, like all things, what's that?
Bell-shaped.
L-shaped curve. So you get a big enough population. Over here you got people that can't ride in the back seat. And then you got people over here, can't ride in the car. No matter where. Most people are in the middle. These people could be airline pilots. And these people can go on space programs. They're not gonna get sick even when they don't have gravity. That's why you can go up in space. Does this make sense? But most people are right here. Okay? But anyway, yeah, this is the physiology of Star Tours. Everybody's been on Star Tours?
Yeah.
Or other rides that are like that, where they just show a movie, Cranial Nerve 2. So you go on these little star speeders, right? You think, oh, I'm gonna get to fly through space. No, you're in an elevator. And the elevator has 3 hydraulic lifts, 1 in the front, 2 in the back. And if you look at this picture that's behind this, if you just tilt your head down, it is the same as when you press on the brake. And that's exactly what happens when that star speeder comes flying into the space station. It shows a 3D movie of rapid deceleration, and the elevator just does this. The ololithic membrane sloops forward. Since your brains see and feel nothing, it is putting together these action potentials to give you the sensation of rapid deceleration. You never left the building. You never left the elevator. Any questions? All right. Semicircular canals, we call this dynamic equilibrium. Again, these terms are oversimplified misnomers. In this case, this is talking about turns or rotations. And in the physics of a turn, you are in a constant deceleration in one vector and in an acceleration in another vector. That's what a turn is. So since you have this combination of deceleration, acceleration, they call it dynamic equilibrium. It's about turning. Again, each side has 3 semicircular canals. They are arranged 90 degrees to each other in 3 dimensions, so that way your brain can get information of any gyroscopic rotation in any gyroscopic plane. Everybody got it?
Mm-hmm.
Do you understand what that means?
Yeah,
Do you?
I think.
When 3/4 of the room looks like this, I should lecture like that.
Oh my God.
This is how much circulation the cilia of the hair cells
Fuck.
are embedded in the cupula.
Do we look that dead?
Uh-huh.
Oh my God.
Some of you look worse. I kid you not. You gotta stand here and see it.
Oh God. Take a picture.
At the base of each semicircular canal. The tube swells. Wherever there's a tube that swells, we call that the ampulla. Inside the ampulla is the crista ampularis.
Oh my God.
Hair cells are in the crista ampularis. then the cilia of these hair cells are embedded in this gelatinous cell.
Oh my god.
We call it cupula. Everybody see the hairs, the cilia embedded in the cupula? Endolength moves the cell,
Yes.
okay? And again, it's all about altering resting action potential tone. This is that resting action potential tone. You can either slow it up or accelerate it. Everybody see that? And for Monday Lab, we did this experiment, this demonstration. Wednesday Lab, you'll do it this afternoon, on vestibular nystagmus. It is a complex thing where from 5 different sources, from both sides of your head. How do I know it's 5? 3 are semicircular canals. Utricle, sacral.
Oh, right.
3 semicircular canals, utricle, sacrals. You're getting cranial nerve 8 input from 5 sources, both sides of your head. It's going into your brain. It's going to match up with inputs from the superior colliculi. It's going to match up with several different cranial nerve nuclei. 3, 4, and 6. 3, 4, and 6 are all about moving your eyeballs. And this allows you to track a moving object even though you have a different speed and direction. You can still follow a moving object because of vestibular nystagmus. That's the relevance of it. Do you want to actually talk about these pathways and the various cranial nerves?
Are we required to know it?
No. You want me to talk about it?
Is it interesting?
Not when you look like this.
Okay.
Okay, so I'll explain it again in lab this afternoon, but I got time. Say this is a glass of water. You're at this fancy restaurant where everything's supposed to be perfect. But you can see through the glass that there's something floating on this side of the glass. So you take the glass and you go like this. And the glass moves, and the water don't. So, because there is some friction between the glass and the water, you keep turning it. And because there's friction, eventually the water starts moving. It comes around and goes, oh shit, there's a booger in my water. But when you stop turning the glass, what happens to the water? It keeps going. That is what you want to have in this demonstration we do in lab. When I first spin a student in the stool, the osseous labyrinth is turning like the glass, and the endolymph lags behind like the water. But if I keep turning the student, eventually the endolymph catches up to the osseous labyrinth, and they're moving at the same rate. Everybody got it? So, if I'm turning this way, the cupula goes this way at first, because the endolymph is lagging behind. I spin the student 10 times, and the endolymph catches up, and pretty soon the cupula is straight, which is actually telling the brain, I'm not moving, but you are. What happens when the glass stops moving? The water keeps going, so does the endolymph, and now the cupula bends in the opposite direction. That's the second part up there at the top, which is telling your brain you're moving. From the 8th cranial nerve, bent cilia is telling the brain you're moving, but when the student has the eyes open, 2nd cranial nerve is telling the student I'm not moving, which F's up the brain. And you can see the brain being F'd up in the eyes. Because it's going, 8th cranial nerve, I'm moving, no I'm not. I'm moving, no I'm not. eighth cranial nerve second, eighth cranial nerve second, moving, no I'm not, moving, no I'm not. You can actually see them eye shape that way. Any questions? All right. Great. We could probably get started on the quiz soon.